So as we come to the end of day one of the Fraxa Investigators meeting, I just wanted to note some of the highlights from the breakout sessions. As you can imagine, we have a lot of people at this meeting. We have a lot of really great scientists, and they all want to present their results. We don't have enough time slots for everyone to present their results to the whole audience, so at this point in the day, we go to what we call breakout sessions, where people give smaller group presentations. And it really just depends on the person, whether they're a junior person and they just feel more comfortable in a smaller group, or whether they have a topic that's perhaps of more limited interest so that not everyone in the group wants to hear about this. We try to organize them into these breakout sessions, but a lot of these are also really important research findings. So just to highlight some of the things that were presented there, Doug Ethel from the University of California, Riverside, part of the husband and wife couple that made this breakthrough finding that minocycline is therapeutic in Fragile X. Well, he's also been dabbling in making induced pluripotent stem cells, and it's an exciting new technology where you can take any cell, like a skin cell, for example, or a blood cell, and turn it into stem cells. So you take adult cells and reverse engineer them into stem cells so that you don't have to get embryonic stem cells. And what's especially cool about this in Fragile X is that if you want to do your research on human cells with Fragile X, which most likely are going to be better than working on mouse cells, you can get human cells from a huge number of subjects by just getting some blood samples or skin samples or buccal scrapings, just scraping inside the lining of the mouth. It's really easy to get live cells and turn them into stem cells using this technique once you've mastered it. It's a little bit difficult technically, and it's an evolving technology, but Doug seems to have mastered it. He was talking about some of the different cell lines. So one of the things that we're contemplating at this point is coming up with essentially a stem cell bank where you can take these pluripotent stem cells and distribute them as a research tool the same way we distribute the mouse model for Fragile X. So that was very exciting. We also had a new researcher who came all the way from Germany, Stefan Kindler, presented his findings. He's already got preliminary data talking about the way Fragile X protein regulates the scaffold, the skeleton of the neuron. And it's important when you're talking about neurons because the shape regulates the function and vice versa. So the cytoskeleton, or the scaffold, as they often call it, regulates the function of the neuron. And he's found some very specific ways in which that occurs, and these could lead to alternative treatment approaches. It's always nice to hear from some new folks. Mary McKenna is another Fragile X researcher, another Fraxa researcher, who's looking at metabolism in Fragile X cells, and in particular how neurotransmitters are synthesized and metabolized, and finding that there's an enormously increased rate of turnover in Fragile X cells, which tends to lend credence to this notion that there's just too much excitatory neurotransmission going on. We also had a postdoctoral fellow funded by Fraxa from Julius Jou's lab at the University of Virginia, talking about ways to rescue synaptic plasticity in the knockout mouse using a number of available drugs that work on the serotonin system. And this is a nice kind of translational pharmacology that gives us pointers as to which way we can treat people with Fragile X that might actually enhance the outcome in the long run. So potentially disease-modifying approaches that utilize available drugs. So things like Abilify and other drugs that block certain kinds of serotonin receptors could be very helpful, especially beneficial in Fragile X, one of the key findings from this. Also heard some additional talks from the fruit fly researchers that made us think that the fly is, in fact, much better than anybody thought as a model for studying Fragile X, and some very encouraging results from Makis Skoulakis, who's a Greek researcher in Athens funded by Fraxa, and he's got some great results from fruit flies showing that a number of treatment strategies can have specific effects, and he's actually refining the protocols to make it even more efficient by using the fruit flies, for example, that just have one copy of the gene knocked out, and he actually gets phenotypes in these flies which can be rescued with drugs. One of the interesting things that he actually reported on was that in some of the so-called vehicle-treated flies, he saw therapeutic effects. And the vehicle is just the stuff that you put the active drug in, but in his case the vehicle was ethanol. And so he found that in fruit flies, maybe having a drink could in fact benefit people with Fragile X, or at least flies with Fragile X. I guess this is another kind of cocktail approach to treating Fragile X, literally with a cocktail. Following that, we also heard some interesting results from Delia Kruger in Mark Baer's lab, and she's developed a very nice cognitive assay in the knockout mouse for measuring changes in learning and memory. It's not exactly quick or easy. First of all, you need mice, and they're hard to breed and expensive to keep, and the protocol requires quite a bit of training, but they've gotten it automated, and they've managed to show that you can actually reverse some of the intellectual deficits in the mouse using doses of MPAP research type treatments that you can give to an animal. So that was pretty cool, and obviously that's something of interest to further research going forward. And during the start of the second day, we had a lot of people talking about the effects of Fragile X on synaptic function in particular. As you can imagine, that's something that a lot of our researchers are working on. So a lot of folks are looking at how the loss of the Fragile X protein affects the synapse. Justin Fallon talked about how the loss of the Fragile X protein affects the axon part of the synapse, and what FMRP might be doing presynaptically. So it's not in the dendrites, which is what we've been talking about all along. When we talk about this MLUR theory and increased long-term depression, Justin's actually looking on the other side of the synapse and finding that Fragile X protein is probably important there too. It's definitely very important on the presynaptic side in development, but it's probably also got an ongoing function every day in the axon as well. It's not surprising because the two halves of the synapse talk to one another, and the signaling is not all in one direction. So the dendrite actually talks back to the axon when the signal gets sent, and I suspect that a lot of that communication is moderated by FMRP. Exactly how that's going to affect treatment at this point, we're not sure, but we're keeping a close watch on this. Kendall Brody presented some more interesting fly data where he looked at the relative functions of FMRP along with the proteins produced by the homologous genes FXR1 and FXR2. And it was a very elegant series of experiments designed to show essentially what the specific functions of the different genes were. And the upshot of it was that, at least in flies, you could actually reproduce most of the function of the fly gene just by substituting the human FMR1. So this just kind of lends a little bit more credence, as if we needed it, to the fly model, and that it's actually a pretty good way of modeling the absence of FMR protein in humans.